Hello, my name is Professor Bum-Young Liu from the Jungang University, Department of Animal Science and Technology, South Korea. I would like to thank for the World Journal of Mental Health for allowing us to present our paper.
Title of paper is Bisphenol Analogs Downregulate the Self-Renewal Potential of Spermatogonial Stem Cells. In recent years, the escalating use of plastic has led to increased human exposure to bisphenol, including BPA and its substitutes. This heightened exposure has raised concerns about potential health risks, particularly in the context of male fertility. Despite numerous studies revealing BPA's toxicity, our comprehension of a BPA substitute remains limited in male fertility. Therefore, this study aimed to investigate the effects of BPS and BPF in comparison to BPA on the self-renewal potential alteration of spermatogonial stem cells, which are a fundamental source of spermatogenesis and male fertility. In this study, mouse spermatogonial stem cell, abbreviated as SSC, were exposed to bisphenol analog with different concentration for 7 days in vitro culture. And a comprehensive approach to assess the effects of a BPA substitute on SSC population. Trypan assay was used to assess cell viability and to quantify live cell number after bisphenol treatment. Apoptosis analysis using annexin-5 and flow cytometry, Western blot immunocytochemistry, and QRT-PCR were performed to evaluate the gene expression patterns. And finally, the functional alteration of SSC was investigated by examining donor SSC-derived spermatogenicity evaluation through in vivo transplantation followed by testis analysis. As a result, BPF exhibited a similar inhibitory effects on SSC as BPA, demonstrating a significant decrease in SSC survival, inhibition of proliferation, and induction of apoptosis. On the other hand, while BPS was comparatively weaker than BPA and F, it still showed significant SSC cytotoxicity. This slide illustrates the restoration of spermatogenesis driven by donor SSC, visualized through GFP expression. A single donor SSC can self-renew and differentiate former germ cell colony within the recipient seminiferous tubule. Therefore, GFP-guided colony counting analysis serves as a valuable tool for directly and quantitatively evaluating the presence and function of SSC introduced into the testis from the donor cell population. The number of colonies formed per 100,000 SSC, representing stem cell concentration, was significantly diminished at a concentration of 100 micromolar for all bisphenol treatment group compared to the control. Notably, the total number of colonies exhibited a clear dose-dependent decrease with a significant reduction observed from concentration of 12.5 micromolar for BPA and 25 micromolar for both BPF and BPS. Upon intergroup comparison, a substantial distinction in total colony count was observed, indicating that BPA, BPF, BPS hinder SSC proliferation, subsequently diminishing the counts of functionally active SSC within the population, thus impacting stem cell number. Based on our experimental result, We believe that further research and regulation are necessary to ensure the safety of these common bisphenol analogs. Thank you for your time and interest in our work.